Hello, at Medical Sciences Made Easy by Naphtali Muhumuza. And in this session, we want to look at hemoglobin synthesis. In the previous video, we looked at the structure of hemoglobin, whereby we said hemoglobin, that if you have hemoglobin, is made up of heme and globin. And is made up of heme with globin. These globin are made up of 4 polypeptide chain, that is 2 alpha and 2 beta for the adult hemoglobin. And for the fetal hemoglobin, we see 2 alpha and 2 delta. Then for HbA2, we see 2 alpha and 2 gamma. Actually, for A2 is 2 alpha and 2 delta. Then for HbF is 2 alpha and 2 gamma. So these are the polypeptide chain together with the heme group, what we call heme prosthetic group. And we saw that these polypeptide chain are made up of 141 to 146 amino acids. And now today we want to look at how can we manufacture this hemoglobin made up of heme and globin chains. So the first thing we're going to look at is the synthesis of heme. Then we shall wrap up with synthesis of globin. Then we combine them to form hemoglobin. And as we know, then we shall conclude by looking at the functions of hemoglobin in the body. So by the end of this session, we should be able to understand how heme is manufactured, how globin is synthesized, and how do we combine them to form hemoglobin. And we shall also need to know the structure, know the functions of hemoglobin in the body. And of course, as we get started, always remember to subscribe, like the video if you learn, and always share your comments and share to many others. So we are beginning with number 1, which is heme synthesis. And this heme, which we normally see that when we have heme, it is composed of the porphyrin ring and iron in ferrous state. So we are going to see the first how porphyrin ring is formed. After knowing how the porphyrin ring is formed, we shall see how iron is incorporated within the ring to form heme. And after seeing this, heme starts with the first step occurs in the mitochondria. This is the mitochondria. And within the mitochondrion, we're going to see the succinyl-CoA, or what we call the succinyl-CoA. The succinyl coenzyme A, even we shall see the incoming of glycine. And this succinyl-CoA and the glycine combine together. After combining together, they form a compound known as delta aminolevulinic acid. It is called delta aminolevulinic acid. That is what we call delta AL. We abbreviate it as AL. And this reaction, whereby the succinyl-CoA within the mitochondria, because it is produced in the Krebs cycle, it is produced from the Krebs cycle. Then we see glycine entering into the mitochondria, combining with this succinyl-CoA, enzyme A, to form delta aminolevulinic acid in the presence of an enzyme known as delta aminolevulinic acid synthase. So it is catalyzed by a synthase, and synthases are named according to the products they are forming. So this is the first step in heme synthesis. The first step in heme synthesis is the formation of delta aminolevulinic acid, which occurs in the mitochondria in the presence of delta aminolevulinic acid synthase, which combines glycine together with succinyl-CoA enzyme. And after forming this delta aminolevulinic acid, it is moved in the cytosol. This is the cytosol side. So it crosses the mitochondrial membrane and enters into the cytosol. And within the cytosol, we shall see two molecules of delta aminorevenic acid combining to form fibrinogen, combines to form fibrinogen. Two molecules of delta aminolevenic acid combine to form porphobrinogen in the presence of porphobrinogen synthase, dehydratase. Here we have delta aminolevenic acid dehydratase, meaning it is removing water. There is removal or loss of water molecule to form porphobrinogen. So two molecules of delta aminolevenic acid combine together by loss of water to form porphobrinogen in presence of delta aminolevenic acid dehydratase. Dehydratase meaning we are removing water molecule to form porphobrinogen, and this porphobrinogen formed four molecules of them. The four molecules of porphobrinogen combine to form an intermediate known as hydroxymethylbilane. Bilane is an intermediate which is a linear tetrapyrrole ring. It is a linear tetrapyrrole. So it has four pyrrole rings. This is hydroxymethylbilane, which is an intermediate, and it is formed by the reaction of the enzyme that catalyzes the reaction is called porphobrinogen deaminase. So we see porphobrinogen deaminase enzyme combining four molecules of porphobrinogen, forming hydroxymethylbilane, which is a linear tetrapyrrole ring. So it is still in linear form, it hasn't formed a ring. So it is forming a linear tetrapyrrole. It is still linear, it has not formed the ring. And this hydroxymethylbilane is the one that is converted to uroporphobrinogen 1. And this conversion of uroporphobrinogen 1 directly from hydroxymethylbilane to uroporphobrinogen type 1, it is catalyzed by an enzyme known as, it is a synthase, and the synthases are named according to the substrate they are forming. So it is uroporphobrinogen synthase 1. Synthase type 1, which catalyzes the conversion of hydroxymethylbilane to form uroporphobrinogen type 1. This uroporphobrinogen type 1 is still a linear structure, it hasn't formed a ring. And then this uroporphobrinogen 1 is converted to uroporphobrinogen 3 by a co-synthase, and this synthase is also known as uroporphobrinogen synthase type 3. It's the one that converts uroporphobrinogen type 1 to uroporphobrinogen type 3. And after forming uroporphobrinogen type 3, which is still a linear structure, it is now at this point whereby we are going to convert a linear structure of a tetrapyrrole into a ringed structure. And this one involves loss. We are going to see loss of 4 carbon dioxide molecules. We are losing 4 carbon dioxide molecules. So meaning this process is decarboxylation. Decarboxylation because we are removing carbon dioxide. And the enzyme that is doing that is uroporphobrinogen decarboxylase. This uroporphobrinogen decarboxylase enzyme is the one that cleaves off 4 molecules of carbon dioxide from uroporphobrinogen type 3 to form coproporphobrinogen type 3. And this coproporphobrinogen type 3 formed is a ringed structure. So it has formed already the ring. And this coproporphobrinogen type 3 is the last reaction in the cytosol of the cell. After forming this ringed structure, it is taken back into the mitochondria. It's taken back into the mitochondria, whereby this ringed form structure is converted to protopophyinogen 9. So we are seeing conversion of coprophosphobilinogen 3 to protopophyinogen 9. So this protopophyinogen 9 formed is by the reaction known as oxidase, it undergoes oxidation reaction by an oxidase enzyme called coprophosphobilinogen 3 oxidase to form protopophyinogen 9. And this protopophyinogen 9, which is still a ring, the form, it is a ring form, and our major aim to form porphyrin ring. So it is going to be still further oxidized. That undergoes an oxidation by an oxidase to form protopophyrin 9, which we normally call porphyrin ring. So this is, you can also always call it porphyrin ring, or you can call it protopophyrin 9, which is formed in the presence of an enzyme oxidase, whereby this oxidase is oxidizing by adding oxygen molecule to protopophyinogen 9 to form protopophyrin 9, which is a ring form. You can call it porphyrin ring. Now, since we have formed the porphyrin ring, what we are remaining is to incorporate the iron 2 in the iron in ferrous state. So what happens is that the bone marrow keeps the iron, and what happens here, we see iron coming in ferrous state and is incorporated in this protopophyrin ring 9 to form heme, and the enzyme that incorporates iron into the ring is known as the ferrochelatase. So ferrochelatase enzyme, ferrochelatase is the one that brings the iron in ferrous state and adds heme at the center of protopophyrin 9 to form our darling heme. So here we have the heme after ferrochelatase coming and adding iron at the center. So we shall see if we have this ringed form, it will put iron at the center. This iron at the center is surrounded by the pyrrole rings. And these 4 pyrrole rings are the one that we shall see them form. So we form the pyrrole rings, whereby here we form another ring. Here we form another ring. And here we form another pyrrole ring. And these 4 pyrrole rings are joined together by the methane group. We join them together by the methane group. So we have another pentose here, another pentose here, another pentose here. So without iron, we are saying this is protopophyrin 9, or we are calling it porphyrin ring. So ferrochelatase, his role is to bring the iron and adds heme at the center. He puts him at the center in ferrous state, and this is what we call heme. This is heme, which has been formed, whereby this heme, it has the 4 pyrrole rings. It has the four pyrrole rings, which we are talking about. So after forming the heme, both we have to know in this process the stages that occur in the mitochondria and the stages that occur in the cytosol, because we can easily ask you to outline the steps that occur in the cytosol or the steps that occur in the mitochondria. And we have seen the mitochondria is the first stage and the last 3 stages, whereby this is stage number 1, number 2, step number 3, step number 4, number 5, then number 6, number 7, and number 9. So we are seeing step 1, 6, 7, and 9 occurring in the mitochondria, and step number 2, 3, 4, and 5 occurring in the cytosol of the cells. And this is what we call heme synthesis. After we have synthesized the heme, now we need to synthesize the globin chain, or what we call the polypeptide chain. Because at the long run, hemoglobin is formed of heme and the globin. So after synthesizing this, we are going to see the synthesis of heme, of globin. And this synthesis of globin, for it is not so much hectic here, that globin is synthesized in the cytoplasm in the presence of ribosomes. So it occurs in the cytosol, whereby the ribosomes act as machinery. That when we have the DNA in the cell, if this is the cell, we have the nucleus with the DNA. What happens in this cell is that the DNA undergoes replication to form two daughter cells, which each has a template as the mother strand. Then after replicating, it forms a transcript. And this transcript, which is formed from DNA, is now which is known as mRNA, messenger RNA. And after forming messenger RNA, it exits from the nucleus into the cytosol, where we find ribosomes, whereby ribosomes have the larger subunit and the smaller subunit. So this, we are seeing mRNA exiting the nucleus, coming into the machinery which we call ribosome. So this is the ribosome, and it is in the presence of ribosome together with transfer RNA, whereby transfer RNA brings the anticodons complementary to the codons of the messenger RNA to form a polypeptide chain. So we are going to see within the ribosome, there is interaction of mRNA, that is messenger RNA from the nucleus, which is carrying the codons, that is triplet of codes, whereby adenine, cytosine, guanine, and uracil, whereas we shall also see the action of transfer RNA carrying the anticodons. It is also a triplet of codes, and these guys must be complementary, and they are the ones that command which type of amino acid to be brought. So we are going to see the transfer RNA. For them, their role is to bring, they are the ones that transfer the amino acid. The amino acid, which is complementary, can be coded before by the codons on the mRNA. And when this transfer RNA is charged with the amino acids, come and in the presence of ribosome, is able to form a polypeptide chain. These are amino acids, different amino acids, and whenever we reach 141 to 146, it is terminated. So we see continuous manufacturing of a polypeptide chain, transfer RNA bringing the right amino acid depending on the codon read by the anticodon of the transfer, until we form a long polypeptide chain, being ribosome being the machinery. And after forming a polypeptide chain, this one we are calling it a polypeptide chain formed. This polypeptide chain has amino acids ranging from 141 to 146, and after being formed, these amino acids, it undergoes, this polypeptide chain undergoes configuration. It is configured, or it is folded. It undergoes folding to form it is the one that results into 4 globin chains. The one that results into 4 globin chains, whereby these 4 globin chains, they can either be alpha, they can be beta, they can be delta, they can be gamma. depending on the type of normal hemoglobin, whereby we said if it is HbA, it has 2 alpha and 2 beta. If it is HbA2, it has 2 alpha and 2 delta. If it is HbF, that is the fetal hemoglobin, it has 2 alpha and 2 gamma. So this globin chain depends on the type of the hemoglobin which is being synthesized. After being folded, the last part is combining the heme which is formed. So the last part is that the heme which is formed exits the mitochondria. And after exiting the mitochondria, we form the 4 heme groups. And these 4 of the heme are the one that goes and combines with the 4 of the globin, either alpha, beta, or gamma. And their combination results into formation of hemoglobin. So hemoglobin, at the end of the session, we're going to see that hemoglobin has 4 heme groups, and each 4 heme group is attached to a single polypeptide globin chain. And because the globin chains are 4, even heme are 4. So by the end of the session, we're going to see formation of hemoglobin with how many? 4 heme groups and 4 globin. And these globin are polypeptide chain made up of 141 to 146 amino acids. So this is the synthesis of heme, whereby it is in 3 stages, which begins with the synthesis of heme, followed by synthesis of globin, which is number 2, then followed by combining the 2, which is step number 4, whereby we combine heme together with globin, 4 of them, to form our fully functioning hemoglobin. And after forming this hemoglobin, we know it is very important in our body. That this hemoglobin, which is formed in both in the mitochondria and the cytosol, these reactions we have seen, this hemoglobin plays a very pivotal role in the body, whereby the functions of the hemoglobin, and look at the functions of the hemoglobin which we have formed. But number 1, they are responsible for transport of oxygen from lungs to respiring tissues or respiring cells, stroke tissues. That is one of the functions. Another function is that they transport carbon dioxide also from tissues, respiring tissues or cells, to the lungs for exhalation. So whenever you want to breathe out carbon dioxide, it is the hemoglobin that brings the carbon dioxide and also takes the oxygen. And what enables this hemoglobin to deliver oxygen to respiring cells is an important subject known as 2,3-DPG. 2,3-diphosphoglycerate, or B for some books put B. It is an important substance that lowers the hemoglobin level for oxygen and enables delivery of oxygen to respiring tissues. So oxygen is able to be delivered to respiring cells due to presence of 2,3-diphosphoglycerate, which is an intermediate of glycolysis that lowers the affinity of hemoglobin for oxygen, and then the oxygen is able to be delivered to the cells. That is the significance of this, which is seen in glycolysis. Then carbon dioxide is also brought to the lungs to be expelled. Another function of hemoglobin is that it acts as a body buffer, it acts as a blood buffer. How? By maintaining pH of blood between 7.35 to 7.45. That is hemoglobin acting as a buffer by maintaining blood pH. And lastly, about the functions of hemoglobin is that they have receptors on the cell, that whenever you have an RBC, containing hemoglobin. This hemoglobin or these red cells, but which this one is more of the red cells which have the antigens. So this one is more of the red cells, not the hemoglobin, whereby they determine the blood group of an individual. But this one is more of the RBCs. So the major three functions of hemoglobin is transporting of oxygen, respiring cells, then bringing of the carbon dioxide to the lungs for exhalation, and acting as the body buffer. So this is what we can discuss in this session, whereby we have seen how hemoglobin is synthesized in the body by first synthesizing heme, and the actions of heme reactions we have seen they occur both in the mitochondria and the cytosol. And after forming a ringed structure of heme composing of iron in ferrous state and the porphyrin ring, what we have called protoporphyrin 9, then we form the globin in the cytosol in the presence of ribosomes, which act as the machinery for protein synthesis until we form 141 to 146 polypeptide chain composed of those amino acids, which we have seen that the globin forms, they can differ depending on the type of the hemoglobin. And these globin chains, which are a polypeptide chain, each one combined with its own heme. So we see 4 hemes being formed, combining with 4 globin chains to form the hemoglobin, which carries all these functions we will see. Thank you so much for watching until the end. Always remind you to subscribe when you have learned and share your comments for the betterment of learning. Thank you so much.